Dear colleagues and friends, I really hope you had a good summer and welcome to the third module of the WEMS Hippocrates webinars. The title of this module is Developmental Neonatal Gastroenterology Clinical Correlations. I leave immediately the floor to the chairman of this module, Professor Joseph Novi. Thank you very much.
Thank you, Professor Perry. I'd like to just introduce to you our first speaker, Professor FLAVIA INDRIO. She is Associate Professor of Pediatrics in Foggia, Italy. She's a Senior Consultant in Pediatric Gastroenterology and Neonatology, member of the e-learning program for ESPGHAN, Secretary of ESPGHAN Working Group for Gut Microbiota, and President of the World Society for Probiotics and Prebiotics. She's also a very good friend, and she has been very instrumental in speaking at various Hippocrates seminars in the past, and a coordinator for several of our previous Hippocrates seminars. And I'd like to thank Flavia for being here, and I'd like to thank all of the participants also for joining us in this very interesting meeting this morning.
Yep. Thank you, Joe, for this nice word, and it's my pleasure to be here. I thank you, and I thank you all the Scientific Society of UNEPS, and of course, Hippocrates Foundation for this kind of invitation, and hope that the screen is fine for everybody and you can see the presentation. As Joe already and Corrado Moretti told you, the topic of today is the development of the gastrointestinal tract and clinical issue related to motility. These are my disclosure, and these are the lecture outlines. So I will try to give you some information about the development of digestion and absorption. We will focus a little bit more in detail on the intestinal motility. We will go through all the possible role of microbiota in this, and then at the end of the lecture, we will try to give a connection of all this information coming from the science, especially from the basic science, to report them in our clinical practice and the clinical significance. So all of you know that the intestine is a complex organ, and inside of it there are a lot of functions. So it's a very complex lecture that Joe and Virgilio gave to me, because it's not easy to speak function per function, because all the functions of the intestine are strictly correlated with each other. So digestion and absorption of nutrients, they maintain also hydroelectric homeostasis, regulation of peristalsia, that is the most important to my point of view for the nutrition. The modulation of immune response, production of a neuroendocrine signal, and of course, the newest, but not more new, the gut-brain communication. So just focus a little bit on digestion. So digestion, as all of you know, I don't want to be like I'm at university or in the school, just quickly say that digestion is the process of changing food in the simple components that the body can absorb. So in the gastrointestinal tract, there is a place of this digestion and absorption, and this starts from the mouth in different parts, esophagus, stomach, small intestine. So absorption is, we have three kinds of absorption. We have a simple diffusion, a facilitated diffusion, and an active transport. I won't go through this physiology. All of you are familiar. All of you very well know. So the transport and the absorption are extremely important, because this happens in the small intestine, where there are the villi and the microvilli. And this just allows the intestine to amplify the surface of where this absorption and transportation happen. So of course, for the absorption, for the transport, not only the villi, not only the epithelial cells are needed, but also are absolutely important the development and the maturation of vascular system. Because if we do not have the vessel into the villi and into the enterocyte, all this product that came from the intestine cannot reach the general circulation and the organs that were going through work. And of course, it's important also the lymphatic system, and we will see because different nutrients have different kind of absorption. Of course, the transport of lipids, it's a little bit more complex because they need some proteins that just bind and go through the intestine. But let's go quickly on this physiology and see. This is a very old slide. It's a courtesy of Peter Miller, who was very much involved in the development of intestinal function. And Peter Miller made his research around 1890s. So what he found, and because he did it in Britain, was that the digestion and the absorption basically mature around 30 to 30 second week of gestation. Motility, it's a little bit more complex, even in the maturation, because around the 17 weeks of gestation, the motility starts, even though it's disorganized, and then they start to be a little bit more complex until the 32nd weeks of gestation, where start to appear the migrating motor complex. The migrating motor complex is a pattern of motility that are efficient in move forward the nutrients from the stomach to the duodenum to the intestine. If we do not have the migrating motor complex, so the food remains stopped in the part where it's gone, like in the stomach, in the duodenum. And so Peter Miller said that around 37, 39 gestation weeks, so basically when the newborn is at term, there is a full mature motility. Well, after the study of Peter Miller, and of course, after the study of Caroline Berset, we did also our study. It was a long time ago, more than 20 years ago, and we just were agree about the pattern of motility starting around from 28 to 32 weeks of gestation and reach completely the maturation when the baby was at term. These were studied basically done on the motility of the stomach with the electrogastrography. So we were agree with them. 32 weeks of gestation was the end point, well, was the turning point, let's say, when the baby basically could have the full motility and the bolus could be easily transported through the different gastrointestinal tract. And also this was the gastric emptying, so we checked with electrogastrography and ultrasound the motility, but again, this was 20 years ago, and that was the period when this kind of newborn that we are dealing with now did not exist, did not survive. So who of you are in the field of neonatology for more than 20 years, perfectly are aware of this possibility of surviving that was extremely low when the baby was born, below 27, 28 week of gestation. Now we are dealing with 23, 22nd, 24 gestation week, 500 grams of kilograms. And moreover, we are dealing with this new inhabitants of, well, is not more unknown inhabitants of the intestine, that is the microbiota. Microbiota just colonized the intestine and ruled the intestine. I could say, I know maybe Joe will not agree with me, the microbiota rule all the function of the body. And why is this? Because location basically and mainly starting to the intestine. Intestine has got a complex function, immunity function, motor function, epithelial barrier, all of them correlated into each other and driven by the microbes. And we will try to explain why, because if we have some commensal bacteria, good bacteria, and we call eubiosis, some pathway, biochemical pathway of maturation, some pathway of epigenetic, transcriptomic can take and can lead a way that could be totally different if we have the pathogenic bacteria or the dysbiosis. So keep in mind this complex interaction between the microbes into the intestine, between our microflora and the different microflora that we can find in a preterm baby, in a term baby, in a C-section born baby, or in a vaginal delivered baby, and how this huge and immense pathway of biochemical maturation can happen into the intestine and take the effect also far away. But let's try to understand a little bit more in detail. Sorry, I don't know why it's not working. Okay, just to go straight ahead to the intestine and the microbiota. This is a paper that was published some years ago, but Baker and Summer. What do they find? They found even inside the intestine and inside the crypta and the villi of intestine, different kind of microbes. And why is this? Because every part of the intestine has got a room, every part of the villi. So we go on the top, we found epithelial cells, and we found what? Intestinal permeability. So we have a different colonization at the beginning of the crypta and down in the crypta, because the function of this different part of the intestine, microscopic part of the intestine, are different. So complexity of this interaction growing and growing, and we are knowing more and more about this different communication. So what exactly does the microbiota? It change and decide the gut morphology, including the villus architecture and the crypt depth. The stem cell proliferation, we will have some words about stem cell proliferation, the blood vessel density, the mucus layer properties, and the maturation of the mucosa associated lymphoid tissue that, from my point of view, is the most important, because this modification of the immunity of the gut, it's basically ruling all the other functioning. And of course, the maturation of intestinal motility. Of course, there are a lot of paper. Generally, these scientific papers are performed in germ-free rat, in animal, because it's extremely difficult, you can imagine, to perform the study in preterm. And so this, on the top of this slide, you can see the different vessel density related to different colonization. So if we have a germ-free mice, then we have a very poor intestinal blood proliferation, vessel proliferation. And so it's different when there is more diversity of microbes. And so it's the same for the immunity system. So whenever we have a different colonization, a richness, a stability, and the main words that I would like for you to be familiar with, diversity and richness of this, then we have a more possibility, more chance, to get through a normal functioning and a normal maturation of these two systems. For instance, absorption, transportation. I told you about first absorption and transportation, they go together, but they go together also with the vessel maturation. And so again, here, immunity. And why immunity is so important? Not only for the immunity itself, but also we will see for the maturation of motility, because the immunity, the cells of the immunity are strictly related, we will see, of the one of the motility. Some few words of stem cell. Recently, this stem cell was discovered into the crypt of the intestinal villi. And remember how the microflora can stabilize and define also the depth of the crypt. So intestinal cells, they are able to perform regeneration and proliferation of enterocyte, of muscle cells, of neuronal cells into the intestine. So it's extremely important. And now a lot of studies are going through of this intestinal stem cell related to the microbiota. Just a few study I want to show, for instance, for the epithelial cells and globate cells, and well, all the part, let's say, related to the absorption and transportation. This is a nice study, even if it's of some years ago. What they did was this. They took the feces, the microbiota, from preterm newborn, a group of a preterm newborn with a normal weight gain, assuming that weight gain was, let's say, unhealthy characteristic. And they transformate it, so they transplant this fecal microbiota, this species, into some germ-free rats. And so they did the same with some other feces from newborn with a poor weight gain. And of course, they compare with the germ-free mice. Remember that I told you that the main scientific discovering and scientific research on this microbiota and immunization came from germ-free rats. So what the author find in this three group? So they found basically this. Then the mice, well, the intestine of the mice who were receiving the feces of the well-being of the healthy preterm newborn had a development of globate cell, of villi, of crypts related to the germ-free mice and to the rats who received the feces of the poor weight gain baby. So what this means? That again, you have a stable microbiota, you have a good nutrition, and you have a possibility to going quickly and to going safely and to going directly to a good possibility of maturation. So again, how much is important at the globate cells, epithelial cells. And this is another results that we found in this study, not only the number of the cells, but also the gene expression. It's so important, keep in mind this, because sometimes it's not important only the number of cells, morphology, it's also important functioning of any cells. It's up to the genes, to the expression and transcription of the genes that the cells has got inside its DNA. So this, again, intestinal staining, intestinal specimen of this mouse that received the well-being, I could call well-being preterm, so had a much more better expression of proteins. And so this means that the intestinal barrier was functioning quickly and much more with less time compared to the other. Remember this, in nature, there's nothing that don't follow the route. It's just a matter of time. So what we want is to go quickly in the way. And because what will happen in this way, if we go slow, a lot of external factor could come inside and disrupt the normal function. So let's go focusing a little bit on peristalsia. Again, I hope you forgive me that if I give you some physiology and very well known information about this. Peristalsis of the intestine is due to the two different muscle layers that there are all over the intestine. So muscle layers that are circular and longitudinal, and the contraction and the adequate regulation of this contraction allow the bolus to go through the intestine until the very end. This is a scheme where you can see that these two layers of muscle cells are just put it on the top of a myenteric, a layer that is called submucosal, enteric. There are on the top of this net of interstitial cells of Cahill and neurons. So we have on the top the mucosa, the muscularis mucosa, the submucosa, the two muscles, obliqual and circular, and on the below, on the bottom of the wall of the intestine, we find the neurons. So it's a layer, epithelial, submucosa, and muscles and neurons. Into the submucosa, we still have, look, so these muscles are basically surrounded by neurons on the above and on the top by neurons. Interstitial cells of Cahill that are glial neurons and glial cells, and intestinal neurons, enteric neurons, that are basically neurons. They derive from the crestal, neuronal crestal during the embryonic development. So this scheme goes through all over the intestine. The motility pattern is different in the stomach, is different in the ileum, is different in the colon, just like traces, but the anatomical and the morphology is always the same. This is a scheme where, as I show you in the submucosa, so we have this glial cell, the neurons correlated to the glial cells, interstitial cells of Cahill, and look at this, we have the immune cells. These are the immune cells, strictly in synaptic contact with the neurons and to the glial cells. So this, what happened into the intestine, is called neuroimmune interaction. That's why, remember why I told you, it's extremely important that all the process of maturation will start from the immune maturation of the gut-associated lymphoid tissue. This will be reflected on the neuron, and will be reflected on the motility, will be reflected on the also epithelial cells and the permeability of the intestine. So what do we know about the maturation of these enteric neurons and the motility? This is again another scheme that just reminds the previous one. So again, 22nd, 28, again, all study from Peter Miller. But Peter Miller not only start this study on motility, but also he create a very strong school and in London, where they just continues to working on the development of this enteric nervous system. So they migrate, as I told you, from the neural crest into the gut. So they came from the part of the brain. So they proliferate into the gut. They assembly created the ganglia. So they differentiate and into different enteric neuron types and glial cells, and they start to form the circuit to control the gut motility. Of course, this happened during the fetal life, but we will see about prenatal, postnatal age, I will tell you. And of course, after the birth, still there are some modifications, but basically morphology came during the fetal. This is a very recent, well, not very recent, it's a couple of years ago, the same group of, well, a school of Peter Miller with Alan Burns and Nikhil Thapar. Basically, they study on the embryonic intestine how the enteric neuromusculature in the human and fetal colon start and proceed. So this is embryonic week 12. So they start to appear and they start to organize 14 weeks and at 16 weeks. And look at this. So the network starts again to create a plexus, starting from the 12 weeks and coming until 16. But what is happening also is that at 16 weeks, that start the electrical activity related to the gene expression. So there is a temporal development of a number of different neuronal cells and also the glia between 12 and 16. And at 16 weeks of gestation, we do have also the functioning. Couple of weeks ago, the same group with Alan Burns together with the Hong Kong Center, they define how this migration and formation goes from different part from the crest, neuronal crest into the intestine and then different tract of the intestine, because they start from the beginning, well, from the ileum and then go to the colon. They could stop anywhere, just creating some disease. And so it's a very complex. They identify, even though the protein that are related to this migration, they connect to each other. So every how do you see, step that can be interrupted here can stop this migration. And these are the motility. So what about hormones? I forgot to say that we have a lot of hormones into the intestine. Well, basically, intestine is the neglected endocrine organ. So because a lot of hormones are secreted into the intestine, into the villi, into the crypt, especially into the crypt. One above the other is this, is the serotonin. The serotonin is secreted into the crypt, into the intestine, and serotonin is very well known as one of the most important mood hormones. So it's got action not only on the intestine, but also on the mood. And then start the chapter of gut-brain communication. And what's about this microbiota and communication and affecting this production of serotonin. So there are a lot of studies. This is one of the pioneering studies where it showed that these gut microbes regulate the level of 5-HT, that is the precursor of serotonin. So this precursor is just impacting motility and impacting also the secretion of serotonin. So again, the microbiota rules. And what about the connection of gut-brain communication? This is a recent paper by Rochelis Diaz that's working Karolinska Institute. So they found, the group of Rochelis found these new molecules that is called bacteria peptidoglycans that came through the blood from the intestine to the brain. So not only affecting the maturation of the enteric nerve cells and functioning moreover, but also the brain cell. So mood and behavior, intestinal and gut. So gut feeling, as we also say. So this is an open field of research, and that's why we can talk about this, and microbiota again has an important role on this. So just I didn't talk today about the colonization. So how can we do? What baby? Born sterile, born colonized? Well, for sure, the fetus is born immune suppressed. So baby is immune suppressed because it's needed this immune suppression to stay into the uterus. And that, well, we can discuss if the colonization happened in uterus or not. For sure, there are some arguing on this, but let's assuming that the colonization start at birth. Why so important? Whenever we start whenever we start the colonization, we decide which kind of maturation we want to have to these immune cells, to immune cells of acquired immunity, because we have the innate immunity and that's it. But innate immunity is regulated also from the crosstalk between innate and acquired is regulated by the colonization of the intestine, and what are the possible changing, the possible factor that influence gut colonization in newborn? A lot. Maybe gestation, maybe yes, maybe not. For sure birth, and for sure in infancy. So different colonization rule different maturation of immune system. And remember, neuroimmune interaction. We mature the immune system in a way, we mature the neurons in a way. We mature the immune system in a way, and we can have an impact again on the other gastrointestinal function. So what's happened in our preterm? So colonization of premature infant is absolutely different from the term infants. This has already been settled. This is a study published on Pediatric Research 2018, where you can see the diversity, the richness, the different phyla to be represented in different percentage in the premature infant and in term infants. What this can provoke? This can provoke for sure a dysbiosis, because generally, because of this different, and look at this on the part on the right part, when the baby is extremely premature, even this, we have a totally different colonization that is very moderate premature birth, totally different from the term. So what we're going to have in this preterm, that just born preterm, we don't do anything, they just born preterm, we got already a dysbiosis. We got already a dysbiosis, we have detrimental gut dysfunctioning, a detrimental maturation of the intestinal function, a high risk to develop disease like necrotizing enterocolitis, and so on. So we need to be very careful in curing and prevent these dysbiosis that happen in the preterm. So what we can do, what we have in our hands, for sure what we have is nutrients, is nutrition. How can we feed this baby? So, and they will see, and this is the clinical relevance. We go to the clinical relevance, nutrition and disease. Of course, this is something that we find when we have a premature baby and with the altered maturation of the intestine. So we have a baby with feeding intolerance. Feeding intolerance, all of you are familiar. It's just provoking abdominal distension, gastric residual or emesis, blood in the stool, metabolic acidosis, onset of apneic episodes, loss of stool, temperature instability, and the hyperglycemia. And of course, we have from the feeding intolerance up to the intestinal failure. So we can have the preterm, definitely the extreme premature baby, we can define them as children affected by intestinal failure. So because this mass, gut mass is reducing in functioning below the minimum amount necessary. So we do need to have some intervention on this just to promote this maturation and to make this maturation going in a right direction. So what we can do? Well, this is a slide that say, but you can ask me, well, but you say that maturation start, especially the migration motor complex and the neuronal, and especially the neuronal enteric neuron system start to work at 16 weeks of gestation. So why do we have this premature with insufficient intestine? Well, there is this nice study from Peter Sangil and from the group of Peter Sangil in Denmark. They study the preterm baby and the term baby. What they found was that the level of the enzyme lactase, maltase, sucrase, and also from the aminopeptidase A and the aminopeptidase was different in the preterm baby and in term. And on the preterm baby, this level reached the normal level after 2 weeks of feeding. So he says, okay, we have a different level. So that means that during the fetal life, we still have maturation, but the proper maturation and the functioning, the biochemical functioning came after the birth. So from birth until postnatal day, the velocity is increasing in preterm baby, but decreasing term peak. So that means that they born with a high level, but then they balance until the normal function. So the creep depth increase in both preterm and term, and over time the velus creep ratio do not change in preterm peaks, but decrease in term peaks. And at birth, the velocity and the velus creep in all three regions were lower compared to the preterm. So what is exactly mean this? That birth and postnatal factors not advancing post-conceptional age are key determinants for gut and immune maturation in this moderately preterm newborns. I would say in one word, nutrition has an important role. So how do we feed those babies? How can we help those babies in dysbiosis, in the maturation? No doubt, breast milk. Breast milk is the gold standard of infant nutrition. These are all the benefits you can see here about breast milk. You are very well familiar. Breast milk contains bioactive compounds, contain intestinal epithelial cell, enteric nervous system, mucosal immune cells, stem cells, microbes that helps to prevent the dysbiosis. So no doubt, but even there is a very recent study, this was published a couple of years ago, where there were two groups of children affected by feeding intolerance. One fed with own mother milk, and the other group fed with the donor milk. And even in these two subgroups, the authors find a difference in clinical parameter, especially in growth velocity and in weight and length velocity. But this is most important, different microbiota. Why is this? Because every mother has got the microbes and the nutrients that her baby needs. So of course, the best will do give mother own milk, but this not always is possible. So that's why we are increasing now the breast milk bank, even if it's not the same of mother own milk, especially in colonization, because as you can see, as you can know, the donor milk is pasteurized, so a lot of bacteria are lost. And maybe probiotics, this will be a very hot topic to discuss. This is a paper we published some years ago, where those baby affected by, with preterm, affected by feeding intolerance were supplemented with strain of Reuteri compared to placebo, and there was a better outcome on clinical parameter and a better outcome on motility. So we checked the motility parameter of intestinal, of gastric electrical activity and gastric emptying time, and also all the day of hospitalization and time to reach enteral feeding. So this could be an option we can discuss. So this was the last one. I thank you very much, and I invite you all to this 7th International Congress on Prebiotics in Pediatrics that will be held online. There are a lot of facilities for fellow residents and doctors that are very much interested in the field of probiotics. Please apply for the free registration. Thank you very much.
Thank you very much for that excellent presentation, Flavia. Let me just start out with one question myself here, that there are many things that we do to our lowest birth weight infants. And it's, for example, it's very unusual for us not to give antibiotics to these extremely low birth weight infants. And I'm sure that that has some effect on the subsequent development of the microbiota. At the same time, you mentioned feeding. Sometimes we don't feed some of these babies at all for long periods of time. I think we're starting to get better, and certainly Nick will address some of this tomorrow. But I was wondering about some of your thoughts on that.
Absolutely right. And of course, I didn't mention antibiotics on the effect of microbiota because this is biological rationale, very simple to understand. While you give antibiotics, you disrupt the mucosal colonization, and that's it. There are some studies, I just skipped, there are more than one study, and for sure what is known, and of course is understandable, it's that the dysbiosis in this preterm newborn that got antibiotics is even worse. So if the condition of the preterm is stable, I mean pH, blood pressure, and acidosis, I would feed them. I would feed even with minimal enteral feeding, possibly with breast milk, possibly given some functional food in order to restore, in order to avoid this disruption, this dramatic disruption of antibiotics. I know that it's not always easy. I know that the situation, clinical situation, came much more complex, but we are not to be afraid when the condition, clinical condition of the preterm newborn allow us, again, stability, blood pressure, heart rate, pH. So please keep in mind, breastfeeding never harm any situation.
Thank you. Virgilio, do you have any comments?
Flavia, I mean, I would thank you for this wonderful talk. I learned a lot, and I would like to expand a little bit, if you could expand a little bit more on the clinical application, the clinical intervention, where you see the baby at the highest risk, and what is sort of the next intervention that you would propose on clinical ground. I mean, it's your view or your paper is very clear on motility problems, where an intervention actually was associated with improved motility. Do you see other group of baby at specific risk, whether you are able perhaps to speculate, to suggest plausible and meaningful intervention, even a bit too early? to do that?
Yeah, no. Thank you very much, Virgilio, for this question. But as you teach me, because you are much more time in neonatology, that there's nothing more personalized than the newborn. Yeah. So, I mean, you are the chair of personalized nutrition, and I can say neonatology is the maximum example of personalization. And I would start from the preterm, going through the local center. What do I mean? If I have a center with a high rate of necrotizing enterocolitis, then I would focus on a possible intervention. If I had a center of that got a donor bank milk, then I would be happy to use this one. If I do not have nothing, I mean, everything needs to be individualized, personalized, starting from the newborn, going to the center, unique center. This is the main problem of neonatology. Impossible to create guidelines universally recognized. I would care child by child.
Thank you, Flavia. Perhaps there is another question a bit more on the scientific side also. Do you think that we can, how can we learn from intestinal transcriptomics? Do you think that if we were able to see the transcript of enterocytes or so, we could be able to identify different risk groups or so? Is there any new, new research or any plan you have on this?
Thank you. This is a very tricky, well, not tricky, but very interesting question, more research oriented. And yes, transcriptomic is important. And now we have this metabolomic approach where we can find a product of biochemical happening in the cells. So maybe if we do a study with large number, we identified, for instance, a urinary metabolites of the transcriptomic gene, and we can find, for instance, the baby who are at risk to develop enteral feeding or to develop intestinal, and maybe try to find a possible intervention. We are still at the very beginning of this road, but it's very promising. Very promising, sorry.
So you are in favor also? If you had to look, you would do urinary metabolomics.
Yeah, I really like very much. Still not yet, let's say, clear. Still not yet easy to interpret. Maybe artificial intelligence, some software, some computer that can help the doctor. You know, this is the new way approach of medicine. So artificial intelligence, urinary metabolome, transcriptomic, go directly to the enterocytes proliferation or metabolism.
We would like, I share your view. Urinary, we are thinking to move more from urinary to the fecal or the intestinal or the metabolomics of the intestinal content. But the problem becomes 10 times more difficult, far more metabolites, and so we are a bit hesitant.
No,
could you share your view on this?
Intestine is complex, and you find whatever you find into the intestine is not only bacterial, it's undigested food, it's mucus, it's cells, are immune cells, different kind of cells. In the urine, you have less quantity of resident, let's say, and that would be much more easy. So in my point of view, science should focus now on the urinary metabolomic and try to translate with the help of engineer, with the help of computer, with the help of artificial intelligence, translate it more directly. And the next step maybe will be to purify and identify, for instance, short chain fatty acid metabolites into the feces and go straight ahead to them.
Yeah, I do agree. We have a data bank on that, but it's the mathematics and the statistics can be rather difficult. So yes, thank you for your comment.
Yeah.
Is there any comment from the public or so? Okay.
So there are some questions here. One question, I think this is from Istanbul, Turkey. How does intrauterine growth restriction affect fetal intestinal neuronal innervation and later postnatal motility.
Wait a minute, I'm writing. How does intrauterine growth restriction affect fetal intestinal innervation and later? Okay, thank you. So there are some studies that say that intrauterine growth retardation, because generally is due to vascularization, an impaired vascularization of the placenta, of course. This can create something that can slower the maturation. But after all, there is no final, well, it's not been shown any final lesion to this motility. Only it's a matter of time. They will come slowly after birth. Well, months after birth.
I was wondering if Nick had any comments.
Sorry?
Yeah.
If Nick Embleton had any comments.
Nick.
Hi there. Hi, Flavia. Hi, Joe. Hi, Vitilio.
How are you?
Hi, Nick.
Nice
So
to
glad
see
to
you
see
all.
you.
Nice to see you all.
Nice
And,
beer.
thanks for everybody for joining the seminar. And I don't want to give away too much about what I'm going to talk about tomorrow, but I just wanted to pick up on your comments, Flavia, about you know, where we might be going in the next 5 or 10 years. I think I agree with you that analysis of the stool is incredibly problematic. We've done a couple of studies, large studies, where we have collected tens of thousands of stool samples. It is really, really hard work, both to know what to make of the data, because so many of these compounds, you've no idea what the compounds are. The libraries just don't exist. And then in the preterm babies, we know that metabolites or bacteria that actually appear in the nappy, in the diaper, they're a long way away from the action in the small intestine. You know that as soon as the baby becomes sick, they stop passing stool. Some of the sickest babies, where you think, well, they might have got neck and they may have got an ileus, you just don't have a stool sample to work with. So I think I agree with you that urine is definitely worthy of exploration, and I believe, like you, that in 5-10 years time we might well be using those point of care machines like you have in the airport with pattern recognition that identify, you know, a pattern of 20 or 30 chemicals which tell you that there is an inflammatory process going on, be that early stage neck or some other process. But I do think that that's where we're going. And we won't need to know, as a clinician, because I'm primarily a clinician, not a scientist, I don't really need to know what those 20 or 30 compounds are. I just need to know that that's a pattern that looks unhealthy in the baby, in which case I might jump in and stop the feeds. But equally, or even maybe more important, I might get a pattern that says, do you know what, this looks completely normal. There's nothing there that looks like a bit of gut is unhappy, and therefore you can restart feeds, because undoubtedly what happens clinically is that at 2 o'clock in the morning, the resident or the fellow gets a bit spooked and you stop the feed, and people come in and they leave the baby nil by mouth for 48 hours because they're kind of too scared to restart. So I think sort of reassurance from that is going to be quite useful. But coming back then, sort of to pose the question to you, what do you think we might be doing in 5 or 10 years time to get a better handle then on the gut? I mean, it's a very difficult organ to assess in a baby, isn't it? You can palpate the tummy, you can do an X-ray, you can look to see if it's passed stool. What are we going to be doing directly that might give us more information about how the gut is functioning? Because that's key to most of these babies now. Yeah.
You know what I think, Nick, that we should find, and we should put our strength and our energy, and we do need help again. I'm strongly convinced that this way to approach now with artificial intelligence and computer mechanism, maybe, for instance, I'm just, you know, brainstorming. So we do the gastric residual, ultrasound gastric residual, or we perform Interalia. We correlate it with another metabolites in the urine, or we find another quickly For instance, that will be for sure now much more easy in 5 years to have the fecal microbiome pattern in a quick and less expensive way compared to now. So we put together this, we make a score, and we have, you know, the risk of possibility to develop or not, for instance, feeding intolerance. This is my idea. I mean, I don't know if I will still be there doing research or doing clinical because, you know, but in 10 years, this is how I see intervention, prediction value. I'm strongly convinced that if you got an easy, performable value, even that could be performed not only in the tertiary center, you know, also in a small hospital, you get the red lights open, and you know, open wide your eyes and try to do and to approach differently to this pattern. This is my opinion.
Do you think we will develop interventions that improve motility, or is it just a fact of life that if you're a 28-week X 24-weeker, your motility will always be abnormal?
No, improve motility is, I don't know, so far, no. Maybe have an alert.
And
intervention, I don't know. They are just now knowing how do they migrate the enteric nerve, and they are fantastic. I love that center in London. I mean, they
Flavia,
are fantastic.
do you think there is any indirect sort of, it is a correlate between motility, early onset motility, late onset motility, with some other clinical parameter or so?
Mother clinical parameter?
No, but also the baby also. I mean, I got interested in, you know, in the last couple of years to the patient variability. We sometimes think 24-week is a 24-week, and we assume everything is identical. And the more I read and I try to understand, I find that there are 24-week like 22, and there are 24 like 26, 27, from several aspects of human biology. So, do you have any, could you sort of share with you, is there any variability in terms of motility or any associated factor or covariate that you feel commenting on?
Absolutely, there is. I don't know if you just remember how it's complex, the let's say the migration of this enteral neuron in different part of the intestine. It's necessary only a different transcriptase or a different expression of the genes of this CDH19 or SOX10 that are in the enteric neurons, and you got a different, and you got a variability. So now we are starting to go deep and deep in the mechanism, in the physiological mechanism, in the biological mechanism. So again, of course, there is an individual variability. It's not always the same. That's why, again, once again, neonatology, it's more than an example of individualized medicine, personalized medicine. Absolutely, no doubt on this.
Thank you. I share your view. I'm not.
This is Joe. So I think that what you're trying to get at here is biomarkers of maturity at these different ages, at different gestational ages. So, you know, for example, a 13-year-old teenager can be very well developed or not very well developed, and same thing with a 26-week preterm. And we just need to have some very critical features that we can find, and using some of these artificial intelligence techniques, machine learning, et cetera, and a combination of multiomics and perhaps physiologic features, new forms of electrogastrography, for example, ultrasound, things that we can put together as major features for a particular maturity level. That seems to me where we're going in the future.
This is definitely the case for the lung. And the more I try to read and understand, the more I get convinced that it applies to several body organs.
Yeah,
I share completely your view. Yeah.
Okay.
Any questions?
So, what are your views on mixing breast milk feeds with 20-calorie preterm formula, be it in cases where the mother's supply of milk is insufficient. When is it justified to supplement with formula of similar caloric value of breast milk, given that we now know what we now know about the importance of gut microbiota? So somehow mixing the two, whether it be donor milk and baby's own mother's milk or formula.
So the question is if this will be possible.
Yes.
Yeah, this is possible. I mean,
Is
generally,
it possible and justified? I think
it's possible and justified. The answer is yes, it's possible and justified, because if you have the chance to increase the enteral feeding and you don't have enough breast milk, yes, you put it the formula, and maybe a formula with some functional nutrients inside, depending on the gestational age. So yes, it's justified, and it's, yeah.
I'm going to cover some of this in my talk tomorrow. But I think you're right, Flavia. I mean, if there isn't mum's milk there, you've got to use something else. And if you haven't got donor milk, then you have no choice but to use a formula. And to me, it probably doesn't really matter what formula you choose to use. I don't think there's any good data in favor of any specific composition. The one thing that I wouldn't do is I wouldn't tend to mix mother's milk and formula milk in the same syringe, because formula milk is very carefully composed to ensure solubility of all your minerals and fats and those things. And when you add in mother's own milk with potentially quite a high fat content, I don't know what that necessarily would do once it's mixed with a preterm formula, for example, with lots of calcium and phosphate, and you might just form lots of soaps. I've no idea. So I've always said to people, look, if you haven't got enough mum's milk, then give a syringe of mum's milk and then give a syringe 2-3 hours later or something else, because I think that I don't know that mixing the two together in a syringe is safe. I've seen lots of people do it and it's probably fine. But my own feeling is I wouldn't put the two milks in the same syringe and shake them up at all. And you definitely shouldn't shake mum's milk in any case. So.
One other area that we didn't really discuss very much is some of the medications, prokinetic medications. And there's one question about erythromycin, PO, as a prokinetic. And could you comment on that, Flavia, please?
Yeah, sure. Erythromycin is a prokinetic because it's, how do you say, provoke migrating motor complex. Now the problem are two. First of all, as you can see, MMC, migrating motor complex, do not appear before 32nd weeks of gestation. So whatever you give erythromycin before that day, you don't have the target to use. It's as a receptoral problem. Okay, you don't have the maturation of that enteric nerves to be provoked. And moreover, erythromycin is an antibiotic. So all the problem giving more antibiotics, resistance, and all the same. We have an issue with probiotic because with prokinetic because prokinetic were almost there are almost all of them are in the graves because of side effect. They have been off label and they are not used anymore, even in older children. So this was working. I be sincere, around 30 years ago, I used erythromycin for migrating motor complex, but now it's not possible anymore for this problem, I'll tell you. First of all, because children of 32nd weeks almost now do not have so many problems, moderate and late preterm, and because it's an antibiotics. But the effect is this.
At one point, a drug by the name of cisapride was used fairly commonly in neonatal intensive care. Could you comment on that?
Cisapride, I told you, gastroenterologists love cisapride. And now it's off label all over the world because it was shown to have an effect on electrocardiographic traces. So there were arrhythmia and a lot of problem in EEG. It's not used anymore either in older children, and we are all orphan of cisapride. Definitely, I could say we are orphan of cisapride.
I just make a comment on that, Joe. I mean, I agree with that. I would caution people against using any form of pharmacological agent in this situation, that there are no good controlled trials really showing useful benefits from cisapride or erythromycin in preterm babies. Every drug has the potential for harm. And I would strongly advise against people using it, except in extreme situations. The problem, as Flavia pointed out, is not with a lack of motility, but it's with dysmotility. The problem isn't that the intestines aren't churning away. I mean, we can all sit there and we can watch these abdominal swellings and movement across the abdomen. We can see these intestines desperately trying to pump the milk in the right direction. The problem is dysmotility, isn't it? The fact is that for milk to be pumped down that tube, one bit has to squeeze and the next bit has to relax. And the problem that you have in your preterm baby is that it's all chaotic. And I'm not convinced that a pharmacological agent is going to decrease the chaos. It may actually make you squeeze a bit better, but whether that food will go in the right direction, you want it to be going downwards. Whether it will definitely not put it backwards, I don't know. So this is dysmotility. It's not a lack of motility, it's dysmotility. And I don't believe that a drug is going to cure that.
We need a maturation agent, something that induce maturation, right?
Yeah, definitely.
True. But actually, if we look at all our, lots of us now look after 23, 24 weekers who manage to get onto full feeds on day 10 or day 14, who stay on full feeds forever. I mean, we can do it. We can do it. I accept some babies are unusual, but to me, the issues are really around, what's within our control. None of the complex science and the things that Flavia talked about really is in the control of a clinician. The one thing that is in our control are things like, kangaroo care, skin to skin, and mother's own milk. Those are the things we know make these issues better. That's the thing that we can control. So rather than going around looking for this rare pharmacological agent that you might want to occasionally use, I would focus your effort on doing the obvious thing in front of you, which is to try and make sure that we help the mums give their milk to the babies. Unquestionably, that's the most effective, cost-effective intervention that we have.
Yeah, but you know better than I do that sometimes, mother that deliver a 23rd week's gestation baby, they barely can breathe, they have no milk. So I would also, the policy of increasing and give money to this donor milk bank, this is nice. I mean, but it's so, in Italy, we have only one. In Italy, we have only one. Fabio Mosca has got one, but it was a private donation of 3 million euro. It was a private donation, and they can build the breast milk bank with all the criteria needed.
Yeah, I'll say something about my views on donor milk tomorrow as well. And Joe's written a paper on this that emphasizes that inappropriate knowledge or lack of knowledge about donor milk can actually make things worse. I mean, if you assume that donor milk will do what mother's own milk does, you can end up with a worse off situation. So it is a very complex thing. But yeah, I think one of the points I'll make tomorrow is that an intervention like donor milk is more than simply a biological intervention. It says something about your psychology and behavior and the importance of that when we're thinking about how to nourish these babies. This isn't just cells and metabolites. This is about what people believe, and it's that makes the difference on your neonatal units.
There are a few other questions, but I think that it's time to begin to wind this up. And I want to thank you, Flavia, for this excellent presentation. I think it raised a lot of good questions, and I especially enjoyed the discussion about where we might be going in the future and the personalization aspects. And I look forward to, our further discussions tomorrow and the day after in these areas. So thank you very much, everybody. Have a great day.
Yeah, bye-bye. Thank
Bye.
you.
Bye.
Thank
See you
to
tomorrow.
Bye-bye. See you tomorrow.
Bye.
Bye-bye.
Thank you.